Here we are again with news from the website, prostatewarriors.com.
It's great to be back.
Today, we are really on a mission. We're unpacking the absolute latest updates in prostate cancer research.
Yeah, we've got a massive stack of sources here.
And it covers the full spectrum, from active clinical trials recruiting patients right now, all the way to some, well, really surprising biological discoveries happening in the lab.
And I think the tone for today is really important. We're aiming for optimism, but it's, optimism that is firmly grounded in science.
Right.
We're not here to sell false hope. We're here to look at real data, real mechanisms, and real potential for the warriors out there.
Exactly. No fluff, just the facts. Now, I know our listeners specifically requested that we prioritize the stuff that's happening in the clinic right now.
Right, the practical stuff first.
They want to know about trials.
And we are going to respect that request. So here's our agenda for this deep dive. We're going to start right at the top with clinical trials. We have two major updates there. One involves a very clever way to optimize radioligand therapy, and another is looking at a new type of hormone blocker.
Yeah, after that.
Then, once we've covered those, we'll move into a fascinating retrospective study from the VA about chemotherapy choices. It answers a question doctors wrestle with all the time.
Stick with the old or switch to the new.
Precisely. And then finally, we'll wrap up by looking at some preclinical research, essentially the future tech that's just starting to bubble up.
I love that structure. Let's get right into it. Section 1, clinical trials. And we're kicking things off with something called the ARREST trial.
Yes.
This tackles a major issue with 177 Lu PSMA therapy, which, a lot of people will know by its brand name, Pluvicto.
Right. So let's set the scene. Pluvicto has been a game changer. It's FDA approved. It has proven survival gains. For many men, it works incredibly well.
But, and this is the problem the trial is trying to solve, it doesn't work for everyone. The source here says up to 50% of patients get little benefit. That's a huge number.
It's a very high number. And it usually comes down to what we call cold spots.
Cold spots.
You see, Pluvicto works by finding and attaching to cells that express PSMA prostate-specific membrane antigen. But in advanced cancer, the tumors can be heterogeneous.
Meaning they aren't all the same. They're not just clones of each other.
Exactly, even within the same person. Some lesions might be packed with PSMA so they glow on a scan and absorb the medicine, but other lesions, maybe in the bone, they might have low PSMA expression. They're cold.
And the radioligand just floats right past
It just floats right past. And of course, these are the spots that continue to grow and cause problems later on.
So the ARREST trial is trying to fix these cold spots.
Yeah.
How are they changing the drug itself?
No, they're changing the strategy. It's a hybrid approach. They're combining the radioligand therapy with targeted external beam radiation boosts.
Okay, but they're not just blasting everything right. You said targeted.
This is the key. They're using a smart lesion selection process. They're looking for the troublemakers.
Troublemakers, I like that. So what makes a lesion a troublemaker?
Well, they look for a few things. First, tumors that are getting too little radiation from the drug itself. Second, lesions that are aggressive on a glucose scan but dim on the PSMA scan.
The discordant ones.
Exactly. And third, any spot that's causing pain or is at high risk of fracturing a bone.
So they find these troublemakers, and then they just zap them.
They deliver a single high-precision radiation fraction, about 6 to 12 gray. And it's quick, under 60 minutes.
Just one session.
Just one. But here's where it gets really interesting. It's not just about killing that one tumor with the beam. There's a synergy.
A synergy.
The external radiation actually changes the biology of those cancer cells. It unmasks them.
Unmasks them how?
It temporarily supercharges their PSMA expression. Within hours of that zap, there's an 18% increase in PSMA protein and a 2.6-fold spike in the mRNA.
Whoa, hold on. Let me get this straight. You hit the cold tumor with a beam, and the tumor reacts by putting up more of the very target you need.
Exactly. You're turning a cold spot hot.
That is brilliant. It's like painting a target on the enemy right before you call in the airstrike.
It's a perfect 1-2 punch. You sensitize the resistant areas, then you hit the whole body with the drug.
But does it work? I mean, what does the preclinical data say?
Very strong. In mouse studies, this combo extended survival by 60% compared to radiation alone. The tumors grew 2.7 times slower.
60% is. That's significant.
Michael.
But what about safety? You're adding external radiation to internal radiation. That sounds like a lot of toxicity.
Always a concern, but the early data is reassuring. They haven't seen any excess severe toxicity. The side effects seem to follow the known patterns of Pluvicto and mostly reverse in 4 to 6 weeks.
Okay, that's good to hear.
Plus, the trial is adaptive. If a patient is a complete responder, they actually pause treatment to avoid overtreating them.
That's really smart. Okay, a very promising approach. Let's move to the next trial. This is a drug called Opevesostat.
Yes, this is a phase 3 trial, MK5684004. A very different approach. We're moving away from radiation and looking at the hormonal engine of the cancer.
Opevesostat. The source calls it a first-in-class CYP11A1 inhibitor. That's a mouthful. Can you break that down?
Think of hormone production as an assembly line. Most drugs block the end of the line, or they block the receptor where the hormone is received. Opevesostat blocks the very first step in the factory.
So it just shuts the whole factory down.
It cuts the power to the entire building. By hitting CYP11A1, it eliminates all the fuel sources, what we call ligands, that could possibly activate a mutant androgen receptor.
Which is important because these cancers get smart and find workarounds, right?
They do. They figure out how to make their own fuel. This drug starves them of everything.
But this isn't for everyone, is it? The source mentions a specific mutation.
That is the critical takeaway. This trial is specifically for patients with ARLBD mutations. That's the androgen receptor ligand binding domain.
So this is precision medicine. They're genotyping the patients first.
Correct. And the reason they're doing a phase 3 trial now is because the phase 1/2 data from the C-beta study was just staggering.
I'm looking at that stat right here. In the phase 1/2 data, 73.7% of patients with that mutation got a PSA 50 response. Their PSA dropped by half or more.
Now compare that to the patients without the mutation.
Only 8.7%?
That's 73.7% versus 8.7%. It's night and day.
It really is.
It just shows the power of precision targeting. If you give this drug to the wrong patient, it does almost nothing. But if you give it to the right patient, that 20 to 25% of men with this mutation, it can shut the cancer down.
It works fast, too.
Very fast. Testosterone became undetectable in 87% of patients by week 1.
Wow, by week 1. Are there major side effects to shutting everything down like that?
There is one to watch for: adrenal insufficiency. Your body stops making cortisol, which you need.
That sounds pretty serious.
It can be, but it happened in about 13% of patients, and the source says it's manageable with hydrocortisone replacement. You just take a pill to replace what the drug is blocking.
So for that specific subgroup of patients, this could be a new standard of care.
That's the hope. It's moving us toward treating this disease not as one thing, but as a collection of specific genetic subtypes.
Okay, that's the cutting edge. Let's shift gears now to Section 2 and this retrospective study.
Right. Looking back at data from the Veterans Affairs System, a huge cohort from 2010 to 2023.
And the topic is Docetaxel Rechallenge versus Cabazitaxel. Explain the scenario for us.
So you have a patient who took Docetaxel, a standard chemo, and they did well. They didn't progress on it. Maybe they just finished their cycles. But later, the cancer starts growing again.
The doctor has a choice. Do you go back to the old friend, Docetaxel, or do you switch to the newer drug, Cabazitaxel?
And conventional wisdom often says you should switch. The logic is, well, the cancer has seen Docetaxel, so it must be resistant.
Right. So what did this huge VA study show?
It was actually quite favorable for the old friend. The Docetaxel Rechallenge group had a median survival of 12.3 months.
And Cabazitaxel?
9.6 months.
That's almost a 3-month difference. That's significant.
It is. And the response rates were even more striking. They looked at patients who had a massive PSA decline, 90% or more.
And what did they find?
In the Docetaxel Rechallenge group, 9.8% of patients had that huge drop. In the Cabazitaxel group, it was only 3.0%.
So nearly 10% versus 3%. That suggests if it worked well the first time, the cancer might still be sensitive to it.
Exactly. The implication is that for these specific patients, going back might be better than switching. It challenges that assumption that you always have to move to the next new thing.
That's a really comforting takeaway. Sometimes the tools you've used before are still the best tools in the box.
Of course, we have to be careful. It's retrospective data, but it's a very strong signal.
Absolutely. Okay, we've covered the clinic. Now let's put on our lab coats. Section 3, preclinical research and future tech.
And we have two fascinating topics here. One is a biological discovery about tRNA halves, and the other is a new technology called the ALCO5 platform.
Let's start with tRNA halves. That sounds like something out of science fiction. What are they?
Well, normally, tRNA transfer RNA is a good guy. It helps build proteins. But new research found that in prostate cancer, these tRNAs get split in half.
And they become active. Maliciously active.
Yes. The source uses a great analogy, the car brake.
Okay, walk me through the car brake.
In healthy cells, there's a protein called P21. It's the brake. It tells the cell to slow down, stop dividing so fast.
We like P21. We want the brakes to work.
We do. But these tRNA halves act like thieves. They latch onto a protective protein called Y-box binding protein 1. This Y-box protein is supposed to be the bodyguard for the P21 brake blueprint.
Okay, so the tRNA halves swarm the bodyguard.
They do. They're far more abundant. They completely distract the bodyguard. And while the bodyguard is distracted, the P21 blueprint degrades. It gets destroyed.
The brake line is cut.
The brake line is cut, and the cancer cell speeds out of control. And this happens specifically in hormone-fueled prostate cancers.
So what's the implication? Can we develop drugs to stop these thieves?
That's the goal. This discovery identifies them as a potential new target. If we can stop them from distracting the bodyguard, we could restore the brake.
That's incredible. A completely new handle on the disease mechanism.
It's a target we didn't even know existed a few years ago.
Okay, our final topic. The ALCO5 platform. Smart antibody medicines.
This is an evolution of antibody-drug conjugates, or ADCs. We've talked about these before. They're the guided missiles of cancer therapy.
Right, an antibody finds the cancer and delivers a toxic payload. So what makes ALCO5 special?
The innovation is in the linker, the chemical chain that holds the explosive payload to the missile.
Why does the linker matter so much?
Because traditionally, the chemistry only allowed us to attach certain types of drugs. We were limited in our ammunition.
So we had great missiles, but we couldn't strap certain bombs to them?
Exactly. The ALCO5 platform uses a special linker that lets them attach drugs with an alcohol group.
And we should clarify, that's a chemical structure, right?
Yeah.
Not actual alcohol.
Right, a chemical hydroxyl group. But this is a huge deal because a lot of very potent cancer killers fall into this category. They were previously impossible to attach to antibodies.
So this just opens up a whole new armory of weapons.
It does. It widens what we call the therapeutic window. The linker is very stable in the blood, so it doesn't drop the payload early. It delivers these harsh drugs safely to the tumor, sparing healthy tissue.
Hits the cancer harder with less collateral damage.
That's the goal.
It's amazing. From better radiation strategies to precision hormone blockers to reevaluating chemo, and now these new biological targets and delivery systems.
It really is a comprehensive front. We're seeing a shift from trial and error to predict and precision.
So if you're listening right now, what's the headline?
The headline is that the toolbox is getting deeper and smarter. The understanding of who responds to what, like with the Opevesostat trial, is becoming crucial.
It really does sound like the era of one-size-fits-all is officially over.
It is. We're entering an era of customized warfare against the tumor. The treatment is becoming tailored to you.
It's a lot to take in, but it's undeniably positive momentum. The direction of travel is forward.
Absolutely. The science is moving faster than ever.
Well, that's all the time we have for this deep dive. We hope this breakdown helps you understand the latest news from prostatewarriors.com. It is complex stuff, but knowledge is power.
It certainly is.
To all our listeners, thank you for trusting us to unpack this for you. Stay strong and fight on.